Your class schedule. Remember, we're supposed to have both a lecture and a lab exam today. And the lecture exam obviously is the take home. So I had thought about giving you the rest of the time to go work on your take home. But since you're on campus already, we're going to go ahead and lecture, and then we won't have class on Friday. So haven't finished your take home exam by the day that you have that time to work on it. I figured it would save you the time of having to come onto campus. Or if you're already done, you can sleep in, or you can start studying for finals or whatever. So our last unit is on the digestive system. We've got a couple of lectures on the anatomy. This is the most, we've been dealing with basically just one organ, the kidney, all right? We have a lot of different organs in this particular unit, so you need to keep up with the anatomy. We've got the structures of the mouth, the teeth, and the salivary glands, and the taste buds, the parts of the stomach, the small and large intestine, the liver, and the pancreas. And each of those have their own histology structures. So we have a lot more histology of different organs in this unit than we've had of anything probably since thyroid and endocrine, reproductive, endocrine and reproductive. They are very distinct. The liver doesn't look like the pancreas, all right? Both structure and histology-wise. There are some histology structures that are similar, so salivary glands are similar to pancreas, but obviously the pancreas, as you know from the endocrine system, has islets in it, and the salivary glands do not, okay? So our first couple of lectures are going to go through anatomy, and then we have a physiology lecture next week where we start, we're going to go back to the anatomical structures and go through them again, looking at the enzymes that are released and the hormones that regulate them. So that's the physiology part. And that lab for Tuesday next week is a wet lab where we're going to be digesting lipids. So we're going to take whipping cream and add a powdered litmus powder to it, so it looks like alkaline, kind of a lavender color, and digest it with pancreatic lipase, and look at the characteristics and the requirements for that lipid digestion. Okay. So that's Tuesday's lab. And then Thursday, we will be looking at metabolism, okay? And a little bit more of the physiology, and then our last lecture is 2 weeks from today, on a Tuesday, when we look at the endocrine regulation of digestive. So we started with the endocrine system, now we're going to go back. We're going to look at thyroid hormone again, cortisol from the adrenal gland, insulin and glucagon, and growth hormone. Focusing primarily on how they affect blood levels of fatty acids and amino acids and glucose, our three organic forms of nutrients. So this chart right here is really just a summary of the digestion processes that take place in a schematic of the digestive system. Remember, the digestive tract was created when we had the longitudinal folding of the embryo. So the lining of the gut is the endoderm that was on the innermost layer of that. And so food components, whether they're partially digested or not, are really not actually inside the body. They have not crossed the plasma membrane. So it's still an external environment as far as that is concerned, from mouth to anal opening. So we talk about the various components that are going to occur. And we have two different types of digestion. There is mechanical digestion and chemical digestion. So ingestion itself, ingestion, is just putting food in our mouth, okay? So when we eat, that's another term for ingesting. And then we have mechanical digestion. That takes place in the mouth. Most of us don't do that very much. So most of you don't chew food enough for it to turn into a liquid paste. We chew it enough to get the flavor, and then we're done, okay? Even many times we don't even chew long enough to get the flavor. Obviously, we don't chew very much for chemical digestion to take place. When we start talking about chemical digestion, how many of you have taken, say, crackers without salt, because obviously that's going to mask the flavor, and chewed it long enough for the cracker taste to become sweet? Anybody ever tried that? Okay? That is the use of the enzyme salivary amylase breaking down starches to disaccharides, such as sucrose. Most of us just chew it and swallow it. So it takes at least 5 minutes or longer to give enough chemical reaction time for those enzymes to work. Those are still disaccharides, all right? They're still too large to be absorbed. So we don't absorb very much in the mouth. Maybe alcohol, very little water, because remember what the epithelium lining the mouth is? It's a stratified squamous, so that's a layered epithelium. So water is not fat soluble, so it's going to take it a while to get across. So mechanical digestion is primarily the role of the mouth, its primary role. And also, you can see there's a line coming down here from mechanical digestion to the stomach. Most of the digestive activity that takes place in the stomach is also mechanical. So what we're doing when we talk about mechanical digestion is taking a large piece of something and breaking it down into smaller pieces. We haven't chemically changed the nature. So I was born in Burma, which some of you may know as Myanmar, but I still call Burma. And my parents, I was about 5 or 6 when we left, so I don't remember this really clearly. But when roads were made and the gravel was laid down before the asphalt, the gravel was created by people actually sitting on the side of the road and taking hammers to rocks. And literally handmade, taking big chunks of rock into making it into smaller pieces of rock. That's a form of mechanical breakdown. It's not taking, let's say I had a big rock like this of calcium carbonate that I wanted to digest with hydrochloric acid. And so I fill a bathtub up with hydrochloric acid and I put that big rock in there. So the hydrochloric acid is going to start working on the surface of that rock, but it's going to take a lot longer for that chemical breakdown to occur with that big rock than if I were to pulverize it into a powder first. So what have I changed in mechanical digestion?
Surface area.
Surface area, okay? So that's what we're doing in the mouth and in the stomach with the churning of the stomach is increasing surface area so the actual chemical digestion can occur more quickly. So before, well, I'll remind you of this on Tuesday. This is mechanical digestion again, so we're increasing surface area. Now, chemical digestion is also known as hydrolysis. So what molecule do you think is required?
Water.
Water. All right. So here's the basic, and again, I'll remind you of this next Tuesday. So here's our basic product, whether it's the carbon-carbon of fatty acids, or carbons linking amino acids together, or sugar acids together, or sugar molecules together. We mix that with water. This is a reversible reaction. And that's our product. Okay. So this is hydrolysis, this is chemical digestion, and the various enzymes that we're going to describe on Tuesday, such as pancreatic amylase, or salivary amylase, or lipase, or aminopeptidases, come into this. Notice the enzyme doesn't become part of the reaction, it speeds up the reaction. You can take a piece of meat, sterilize it, put it in a jar of sterile water, and over a 2-week period, hydrolysis would occur to break down the protein of that meat into individual amino acids. That would take a lot longer than we typically need, as we require amino acids for protein building or for fuel. So enzymes increase this rate. This would still happen when all we had was water, right? Enzymes just make it occur faster. Okay, before we actually get into the organs of the digestive system, we are looking at another component of our ventral cavity. So our ventral cavities, remember, were thoracic cavity, abdominal cavity, and pelvic cavity. In the thoracic cavity, we had it was separated into two membranous spaces by the pleura. So we had the pleural cavity, and then surrounding the heart, we had the pericardial cavity. All right? So in the abdominal cavity, created by the longitudinal fusion of our embryo, part of that endoderm formed the mesoderm rather, formed the simple squamous epithelium that becomes the serous membrane lining the abdominal cavity. Remember the key word for this membrane? It's up there. We talked about it first when we did the reproductive system. Remember the peritoneum was draped over the upper portion, the fundus of the uterus, and we called it the perimetrium. All right, so we have the peritoneum. So I'm going to draw a different picture than what we have up here, where it's attached to the body wall. It is the parietal peritoneum, where an organ grows into that sac, again, the fist going into the balloon, like the heart or the lung, where that membrane becomes attached to the surface of the organ. It's identified as the visceral peritoneum. But now we have something different that we didn't have with the pleura and we didn't have with the pericardium. And that is the presence of a double layer of the peritoneum. We again first saw this with the reproductive system. Remember the broad ligament was a double layer of peritoneum, and where it was hanging down from the oviduct, we called it the mesosalpinx. Where it was associated with the ovary, we called it the mesoovarium, and the uterus, the mesometrium, or the rest of the broad ligament. So a double layer of peritoneum that's neither visceral nor parietal, it's not attached to the organ, it's not attached to the abdominal wall, is called a mesentery. That's where we got the meso term, all right? So this is a mesentery. The brown in there is connective tissue. So remember we talked about the superior mesenteric artery and vein, the inferior mesenteric artery and vein. That's why it had that term mesenteric, is because it traveled in the connective tissue between the double layers of peritoneum to get to the small intestine or to get to the large intestine. Now, talking about the kidney, the description of the kidney's location, remember, is retroperitoneum. So the kidney never grows out sufficient to develop a visceral peritoneum. It stays behind the parietal peritoneum, and is considered to be retroperitoneal. Some organs are like the kidney. The pancreas is a retroperitoneal organ. Other components of our digestive tract start out with a visceral peritoneum, but then in embryonic development, our intestines leave the embryo and rotate and then come back in, because there's a large opening where the umbilicus is for that to do that. And the first part of the small intestine, the duodenum, comes in, rotates, and is pushed back up against the parietal peritoneum, and that parietal peritoneum, the posterior portion of the visceral peritoneum is reabsorbed, as is the parietal, and so it becomes retroperitoneal. So this is described as a secondary peritoneal, retroperitoneal. Now, I'm not going to ask you to learn which organs are primary and which organs are secondary retroperitoneal, but I want you to understand what that term means. Okay? So it was within a visceral peritoneum and then became attached to the posterior abdominal wall, and that visceral peritoneum was reabsorbed. As we go through the anatomy, I will remind you of which structures are intra and which are retro, but again, you don't have to know which ones are primary or secondary. So I'm going to come back to this image and I'm going to diagram this one on the board. You have this in the lower portion on the left of page 4. You also have a similar image, I think it's reversed left to right, in the study component of your lab packet. You will see this on the lecture exam, where I'll ask you to identify various components of these. So I'm going to draw this a bit larger on the board and take you through it slowly. So this would be the urinary bladder. We'll make this a female. And the uterus and the rectum. Diaphragm. And just under the diaphragm, we'll have the liver, the stomach, and the transverse colon is going to be cut. This is a sagittal section. So we'll see the transverse colon. Not quite that large. The small intestine is almost 21 feet long. So when we do a transverse section, we're going to have kind of the proximal or distal convoluted tubules. We're going to cut through the small intestine multiple times. And then up here, we're going to have the pancreas, which is posterior to the stomach. And I'm going to leave the kidneys out because they're not midline anyway. And I'm going to leave the duodenum out. We'll talk about that later. Okay. So that's a sagittal view. Now let's diagram in the peritoneal structure. So the peritoneum is, I think I'll do it red. So, first of all, I'm drawing parietal peritoneum, right? So the parietal peritoneum is going to be attached to the inferior surface of the diaphragm. It's going to be attached to the deep surface of your anterior abdominal wall. It's going to partially drape over the bladder. Remember, structures that are in the pelvis don't have a muscle separating them from the abdominal cavity. They just have this peritoneum. It's going to be partially attached to the surface of the uterus and the upper rectum. There's a bare area up here where it's not attached to the diaphragm. It's going to go over the pancreas and the kidney. I've left out the vertebral column in case you wonder why this thing looks weird. All right, so that's parietal peritoneum. And that would be this membrane, the white that you see here. Then it's attached to the surface of the liver. And a mesentery, then a double layer of peritoneum, attaches it to the stomach. Instead of calling this something mesentery, it's given the name lesser omentum. It then becomes parietal peritoneum again as it surrounds the stomach. So this would be a wet, moist surface if you were to look at the stomach, just like the visceral pleura or the visceral pericardium. And then it hangs down, forms a double layer that then comes back on itself. So we actually have 4 layers. Now I'm showing a gap there, but in actuality, much of this is fused together at the lower portion. It comes back, it attaches to the transverse colon. So this is now known as the greater omentum. This is also called, in layman's terms, a fatty apron. It's not attached to the underlying small intestine, so it can be lifted up. and lay back down again, just like an apron. Okay, I'll show you some pictures of it in a moment. Now this is the transverse colon. So they go around it again. Let's go ahead and put on here our label of visceral peritoneum. It doesn't have to be just the stomach, but anytime it's attached to the outer surface of a digestive organ, it's visceral peritoneum. So now we have another mesentery. And this suspends the colon from the posterior abdominal wall. So now we can actually use our meso kind of name. And this is identified as a mesocolon, specifically because it's for the transverse colon, the transverse mesocolon. So even though these other structures don't have the word meso in them, they are still a mesentery. They're still a double layer of peritoneum. Now, it's hard to visualize that from this diagram here, but the small intestine is, again, is almost 21 feet long. So imagine a 20-foot long snake, like a Burmese python or an anaconda, that you've laid out on a sheet of fabric, and you take the two ends of the fabric and lift them up and suspend that snake. All right? I don't have long sleeves on, but let's say that my upper extremity is the snake or the intestine, and my sleeve would be visceral peritoneum, except where I lift it up like this would be the mesentery. Okay? So I've got this 20-foot long snake on a sheet that I lift up, and now I have to attach this 20 feet of sheet or mesentery to my posterior abdominal wall. There's not 20 feet between my diaphragm and my pelvis. I'm not that tall. And so that edge of that sheet, imagine gathering all that together to a space of about 8 to 11 inches. And then attaching that to the posterior abdominal wall. And so that's what this root of the mesentery is. So if we look at this diagram, here's the transverse mesocolon. That's that 21 feet of mesentery attached to the posterior parietal peritoneum along the posterior abdominal wall. So the rest of the small intestine can flare out, all right, like the rolled hem of a skirt, and that is the mesentery proper. So where it's gathered up and attached to the posterior abdominal wall is the root of the mesentery. That's what I'm drawing here. That would be the root of the mesentery. And then it's going to form around each of the intestines, forming a visceral peritoneum around each intestine. But the sheet between it is known as the mesentery proper. So let's take a look at some of these structures. Now I've left off the sigmoid mesocolon. That's the S-shaped portion of the descending colon before it becomes the rectum. We'll talk about that, but I wanted to simplify the diagram up on the board. These are the structures that I would ask you to identify on the lecture exam. Well, you will see a diagram like this on the lecture exam. So I might point to this structure here and say, is this intraperitoneal or retroperitoneal? Or this structure, so this would be retro, this would be intra. Or I might ask you to identify the parietal or visceral peritoneum or the other mesenteric structures. Okay? So there will be a diagram like that with a whole list of all the possible answers, and then 3 or 4 questions from those components. Yes?
So what kind of, is it connective tissue that holds those retroperitoneal structures?
The peritoneum itself, obviously, it's a simple squamous epithelial with loose irregular connective tissue. So that on the posterior abdominal wall, where this goes, it's going to be that's going to attach to the fascia of the muscles of the posterior abdominal wall or the anterior abdominal wall. Where it goes over an organ, it would be attached to the capsule of that organ. It would blend into that, similar to the peritoneum on the quiz with the kidney. It would be attached to the capsule of the kidney. So here's an image from Netter's. So we're looking at the liver. And this, what I'll be touching here, so if we opened up the anterior abdominal wall, let's say someone had a general C-section instead of the bikini one. They needed to get to the uterus really fast, so you cut open the abdominal wall. You would be within the peritoneal cavity. So if you laid a hand on the stomach, you'd be touching visceral peritoneum. And if you turned around and touched the inside surface, deep surface of the anterior abdominal wall, you'd be touching parietal peritoneum, just like we did with the lungs or the heart. Okay. So again, none of these organs are actually in the peritoneal cavity. Like the pleural cavity and the pericardial cavity, the only thing we have in the peritoneal cavity is serous fluid. And again, it's for lubrication as these organs undergo peristalsis and movement. So hanging down from the greater curvature of the stomach, the lower inferior border of the stomach, is the greater omentum. So this is that fatty apron that I talked about. Okay. If we lift it up, we're not going to see the stomach, because remember the posterior part of that greater omentum doesn't go back to the stomach. It comes up and attaches to the colon, the transverse colon. The large intestine or colon is shaped like an upside down U. And so here we're seeing the transverse colon. That would be this portion right here of the transverse colon that you're visualizing when you pick up that mesentery. What do you think this membrane here would be?
The root of the
Transverse mesocolon. So it's going from the colon to the transverse colon to the posterior abdominal wall. So that would be the deep surface here of the transverse mesocolon. Okay. So if we look at our models, here's a piece of section of the small intestine, so that yellow material that's added to half of it would be the greater omentum. And here would be showing the ascending colon, transverse colon, and descending colon. So this structure right here would be the transverse mesocolon. And so what we've done is this would fit under here like this. So in lifting up the greater omentum here, you're looking at that surface. The greater omentum is going to be attached here. So this surface is what you're seeing here, which is the inferior surface here of the transverse mesocolon. So here would be the root of the mesentery. And this would be out in the mesentery proper. And this would be the visceral peritoneum actually attached to the small intestine. All right. Here the root of the mesentery has been cut. This would be the beginning of the jejunum, the second part of our small intestine. This is ascending, transverse, descending sigmoid colon. This is retroperitoneal, this is retroperitoneal. We'll go over that when we get to the colon. Transverse mesocolon and sigmoid colon. So those are freely movable against the posterior abdominal wall. And this ruffle here would be about 20 feet long. All right, because we're missing about a foot of the duodenum. So now the liver, here's the greater omentum again. Between the liver and the lesser curvature of the stomach, we have the lesser omentum. So that would be this membrane here. On the left-hand side, We haven't talked about these structures yet, but you do remember the portal vein in the hepatic artery. Along with those three structures, we have a duct coming from the gallbladder to the small intestine. So all of those are going to be found in the thickened region right here. I can get about 2 or 3 fingers through here, and then I would be in the space between the stomach and the pancreas, and partially